Perfect fit every time. Hello there. Welcome to the wonderful world of REAfinity recombinant antibodies, where flow cytometry experiments have already become reproducible. And I'm here to tell you all about it. Our REAfinity recombinant antibodies are optimized for flow cytometry applications. There are 3 super cool features that will make your life in the lab much easier. REAfinity antibodies are recombinantly engineered proteins. They have a mutated Fc region, and they all belong to the same class of isotypes. Sounds too good to be true, right? Let me break it down for you. Right here is the amino acid chain that makes up our REAfinity antibody. What's so important about this chain is that we know the exact sequence of it. Because we decoded the DNA behind it, we have 2 major advantages. We can produce the exact same antibody time after time, and we can engineer the sequence for optimal performance in flow cytometry. So, how does that work exactly? First, we clone the genetic sequence of antigen-binding regions from traditional hybridoma-derived antibodies with a human IgG1 sequence. Then, the REAfinity DNA is expressed in a mammalian cell line under standardized conditions. And by this, high lot-to-lot consistency can be assured. So you see that we are able to optimize these antibodies for flow cytometry, and now I'll show you the part we engineered. This is the mutated part of the so-called Fc region. When antibodies detect pathogens in the body during an infection, they mark them by binding to them with their specific antigen-binding region. The Fc region of an antibody then serves as a docking station for immune cells, so that they can recognize and eliminate the marked pathogens. It's a very important role of our antibodies during immune responses, but in your flow cytometry experiments, it can be really annoying. It causes the Fc region of your flow antibodies to bind to some immune cells that you don't want to detect, which is commonly known as unspecific background signal. So, how do you get rid of this unspecific signal? What people usually do is to add an Fc receptor blocking reagent which binds to Fc receptors on immune cells. Once blocked, these receptors cannot bind to the Fc region anymore. But this is an additional step in your protocol, which takes more time, and we can do better than that. We took the DNA sequence of these antibodies and genetically engineered the Fc region in a way that the final protein cannot bind to Fc receptors unspecifically anymore. This means you can skip the blocking step altogether, and ta-da! This is what comes out from my friend in the lab. Clean results. When using REAfinity antibodies, there is no background signal, even without the use of FcR blocking reagents. Isn't that what you want? Of course it is. But we didn't stop here. There's another feature that really helps out my buddy in the lab. This region defines the isotype of an antibody. Usually antibodies come from varying sources. Some are produced in rabbit, some in hamster or mouse, which means they have different hosts and therefore different isotypes. This also means that you need matching isotype controls for all those species. When using several antibody panels with individual dye combinations, this can easily add up to dozens of different isotype controls for a single experiment, but not with REAfinity antibodies. Look here, all REAfinity antibodies have the same human IgG1 isotype, which greatly reduces the number of isotype controls required as compared to conventional antibodies. You just need one universal isotype control. You know, the best thing is that with REAfinity recombinant antibodies, your flow cytometry results come out in the same quality over and over again. Because we are in control of the sequence, we know exactly which final products we can expect. That's what I call truly reproducible antibodies. All right, guys, are you ready to switch to our recombinant antibodies? The time has come to change now.